Hi, and welcome to section 4. By now you have completed section 1, 2, and 3. We started off little with our atomic understanding of macromolecules. You're going to see how all of these macromolecules come into place, and we're going to continue building on what we learned in the cell and all this wonderful stuff to understand how do our cells use energy and make energy, and how are we in symbiosis with plants on how they do their thing. So this section is called cellular respiration and photosynthesis. So we do cellular respiration, and it's exactly the way it sounds. When you think of respiration, you're probably thinking taking in oxygen, letting out CO2. That is what respiration is. But the reason you and I breathe is because our cells are breathing. And it's weird to think of it that way. When we talked about the mitochondria last unit in the cell, we talked how it was a powerhouse of the cell. I want you to think of them as the lungs of the cell as well. So they're the ones that are kind of doing that oxygen carbon dioxide exchange, if you will, and all that the oxygen we breathe in gets shipped into the mitochondria, and the mitochondria uses that to do a whole bunch of really cool reactions that we're going to talk about soon. And in the process, it creates a ton of carbon dioxide that we breathe out. So it's a huge revelation when you realize, wait a second, the reason we're breathing is because our mitochondria are doing their job. And that's exactly why, because without the mitochondria doing their job, they're not making energy. And without energy, nothing can move or do their job inside our cells. So we are really dependent upon the mitochondria doing a good job for us. And the other part of this section is photosynthesis. You may have heard of this, of course. It's in plants, happens in the lovely chloroplast. Plants have chloroplasts and mitochondria. They got both. We only have mitochondria. We don't have chloroplasts. If we did, we'd be green. Might be cool, but that's not how we are. So plants kind of do both, and you're going to see how they both feed into each other beautifully, and how we feed into them and they feed into us. So get ready, we're about to start section 4, photosynthesis, cellular respiration. But before we do this, of course, we need some background, because in order to do all of these reactions we're going to learn about in the cell, in the mitochondria and the chloroplast specifically, we need to understand the role of enzymes. Now, enzymes, we've alluded to them a little bit in the past when we did our macromolecule unit on, we did carbohydrates, lipids, proteins, and nucleic acids. We're going to really be focusing on the protein part of this to understand enzymes. It's because enzymes are catalytic proteins. So let's start uncovering this. I'm going to tell you how enzymes work, because enzymes are the chief orchestrator, if you will, in making sure these reactions happen. If we didn't have enzymes, these reactions would take forever to happen, or they probably wouldn't happen at all. So we love enzymes, we need them. Without them, nothing's going on. So let's start with enzymes. And as always, here we go, Roman numeral 1. Enzymes. So enzymes, just brief definition, these are catalytic proteins. Now, if you're not used to the word catalytic, a catalyst has catalytic properties. And so let's define what a catalyst is first. You hear about this word in the chemistry world. And what a catalyst is, it's a chemical that helps lower the amount of energy needed to complete a reaction. And it actually helps speed up the reaction in that respect, because if I'm not having to put in so much energy to get this reaction done, I could get it done a lot faster. So let's quickly define what a catalyst is, and then we'll get back to our outline again. So a catalyst, this is a chemical agent that speeds up a chemical reaction without getting consumed by the reaction, and that's important. So catalysts can take the reagents in a reaction, help them react, spit out the products, and all the time they don't get changed. They just help things along. It's kind of like the in-between person on an assembly line. That's a catalyst. So a catalyst is a chemical agent that speeds up a reaction by lowering the amount of energy required to complete the reaction. So a catalyst is a chemical agent that speeds up a reaction by lowering the amount of energy required to complete the reaction. Now, if an enzyme is a catalytic protein, in this case, we've got to kind of flip our understanding of this now. So an enzyme is a protein that speeds up the reaction by lowering the amount of energy required to complete the reaction. So, and same thing as a catalyst, these enzymes, these proteins, these catalytic proteins do not get consumed in the reaction. They're going to stay there as long as their lifespan is determined by the cell. So graphically, if you need to represent this. So let's do a little graph here really quick. So this would be energy, and this would be reaction course, if you will. So the course of the reaction from reactant to product. So if I'm somewhere along this y-axis for energy, and let's say my reactants are along this level of energy, and they're starting to react. What's going to happen in order for them to react? They have to overcome this inertia, if you will. So as they're reacting, they kind of got to get over the energy hump in order to react to become products over here. So what happens with an enzyme, so this is like a normal course of a reaction. When you have an enzyme involved, here's what happens. You have your reactants, but instead of going over this hump, all they have to do, I'll just draw it in here, all they have to do is just do this. So you have ultimately lowered the amount of energy it takes to complete this reaction. When you were without an enzyme, it took that much energy to get the reaction done. With an enzyme, it only took that much energy. And that's significant because things can't get over the hump. It's kind of like trying to push a refrigerator. It's inertia. You have to put an initial oomph into it in order for it to start moving. Sometimes naturally things don't have enough oomph to get things moving, and that's where the enzyme kicks in. So, and so now this energy difference that you see here, the amount of energy it takes to complete a reaction, this is called the activation energy. And this is the amount of energy required to complete the reaction. So what enzymes essentially do is they lower the activation energy. That is a nice sciencey term if you ever take a multiple choice test for AP or SAT. If you see activation energy and you're talking about enzymes, chances are you could start narrowing down your answer right there. But that's a big deal because what you've basically done is instead of trying to push a refrigerator on a carpet, you put it on, what the enzyme essentially does, it puts the refrigerator on wheels on a tile surface. So it's a whole lot easier to get it done. I hope that makes sense. So that's enzymes and how they essentially, what their definition is. Now let's talk about how they actually operate. So the way they operate is a model we like to call the induced fit model. So here we go. This is going to be big letter A under enzymes. Now we're actually going to get into enzymes. So big letter A is induced fit model. And this is where it gets very beautiful, in my opinion, in the biological world. There's so much beauty in the biological world, but the fact that enzymes are kind of a lock and key and these proteins are made in a specific shape to only accept one thing and stuff like that, that specificity, it's gorgeous. So, big letter A is the induced fit model. All right, so I'm just going to give you an example on how this induced fit model works. So, so for enzymes, their nomenclature that we use for enzymes, a lot of enzymes end in ACE, A-S-E. All molecules that you see that end in ACE are enzymes, but not all enzymes are ACEs. I hope that makes sense. So if you ever see something called something ACE, you have an enzyme on your hands. That's a nice, easy thing to know. So let's take, for example, the enzyme lactase. And usually these enzymes are named according to what they bind to or make or whatever it is. They're very specific on their names. So any scientist that's coming around can be like, oh, if it's lactase, I think it interacts with lactose, which makes sense. So go back to Unit 1 when we talked about sugars and carbohydrates. If you remember, lactose is a disaccharide, two sugar monomers put together. In this case, it's glucose and galactose. Those rings are put together to make lactose, milk sugar, stuff that is in our milk all the time. This is also something that people can't digest for some people because they're lactose intolerant. One of the reasons they're lactose intolerant is because they don't make this enzyme called lactase. Lactase helps break lactose into individual sugar monomers, so our body can use that for energy. Keeping them together actually is very painful for the stomach, and they get stomach issues and all that stuff. So let's talk about how this induced fit model works, and we're going to use lactase as an example. And remember, we're dealing with proteins. And proteins are long amino acid chains that get folded up into a specific shape. And so the shape is dependent upon its function. In this case, I'm just going to, in the biological world, we love to draw proteins in literal geometric shapes that we can draw because the protein structure is actually pretty difficult. But let's take, for example, lactase. So I'm just going to draw something like this. This will be lactase. Here we go. So inside your cells, you need to start digesting some lactose. And so let's say you just took in some lactose. And so lactose, I'm just going to draw it very bare bones. Has your little glycosidic linkage there. There we go. And this is lactose. All right. I need to draw that a little bit. Ooh, I'm dropping pens. Excuse me. So, all right, so you can see. The way I'm trying to draw this is so you can see how this structure can easily fit into that pocket in the enzyme. And that pocket actually has a legitimate name. It's called the active site. So the space where the substance that we want to put into the enzyme fits, this spot right here is called the active site. And I guess that one would also be the active site as well. The thing going into the active site, this is called the substrate. All right, so the substrate goes into the active site. Excellent. So what's going to happen now is that these two are going to come together. And this is where it's always been really interesting for biologists to understand this. They're both floating around in the cell doing their thing, but the shape of lactase is so appealing for lactose to go in and bind that there's an affinity, a desire for lactose to bind into lactase. It's really cool. It's a shape-dependent thing. And so what happens is you actually do get this binding. So lactase binds lactose. I'll try it on there, just upside down. Kind of hard to draw them. So then you get this whole pocket that happens right there. When that happens, what's going to happen is the enzyme, which is a protein. Proteins are very flexible, and once they bind into something, they actually conform. They change shape around what they're binding to. What's happening here with lactase, lactase is having a conformational change to actually hug around lactose. And when you see this happen, this is called the enzyme substrate complex. So enzyme substrate complex. And there's a conformational change that's happening with lactase, and that change is allowing lactose to actually react and break into its individual monomers. So without that enzyme substrate complex change, shape in the enzyme change, the reaction can't occur. So you have to have a perfect fit in order for this to happen well. So once that happens, you actually get this nice little hugging happening here. It's not drawn as a hug, but it's actually happening. Then the reaction occurs, and you release, in this case, the individual monomers of lactose. So here would be lactose, and here would be glucose and galactose. And now lactase is primed and ready to go again. So you get this continuous cycle for lactase. It doesn't get used up. As long as there's lactose present, lactase can work. And so what's happening is this enzyme substrate complex is allowing the reaction to happen. So this whole thing is you get your reaction, and here the products are released. That's the induced fit model, and it's cyclical. As long as you have substrate present, it's going to keep going. And in this case, for lactose and lactase, that interaction, that's dependent on your diet. So how much milk sugar are you bringing in, and how much lactase do you have present in your cells? This is why even with people that aren't lactose intolerant, if you have a whole gallon of ice cream one day, you only have so much lactase present. So you're not going to be able to digest all the lactose in time, so you probably will get a tummy ache for many reasons, but one of them is because you kind of still have a buildup of lactose because it hasn't been able to go through the cycle enough to get it all taken care of and digested for you. So everything in moderation, no gallons of ice cream, once in a while. It's okay. So here we go. So that is the induced fit model. That's how enzymes actually do their action. And the last thing I want to talk about with enzymes, and then we'll actually get to cellular respiration in our next video, is how do we address enzymes in the pharmaceutical world and naturally as well. So this last part, before we get into that too, we got to talk about the factors that affect their activity as well. So this will be big letter B, factors that affect enzyme activity. So factors that affect enzyme activity. All right, this is quick because it's fairly simple. Enzymes are proteins, right? And if you remember, proteins, basically, these are folded amino acid chains, right? So what happens when they're an amino acid chain, we call that their primary structure. That's the most basic structure for a protein. It's not functional. By the time it goes to a secondary structure, a tertiary structure, and then finally a quaternary structure, now you have a functional protein. So proteins are functional in their quaternary structure. And sometimes you'll see this shorthanded with a 4 and a degree sign, a prime sign. Not prime, but a degree sign on there. So that's when they're in their functional state. Now, proteins can get denatured. So the word denaturing, very legitimate biological term that you should be familiar with. Proteins can get denatured, meaning they can go from their quaternary structure, functional shape, down to tertiary, secondary, maybe all the way down to primary structure. So proteins can get denatured, and that's an important word, which means they go from quaternary all the way, perhaps, to primary structure by the following. So there's a few things that can actually affect protein shape. And I want you to think about this for a second. Have you ever noticed when you eat food and you eat something raw, like take broccoli? If you eat raw broccoli, it tastes different than cooked broccoli, steamed broccoli perhaps. Here's why. What's the difference between raw and steamed broccoli? What did you apply to that broccoli to make it different? Heat. So heat, high intensity heat, depending on whatever substance you're talking about, actually takes a protein that's in a specific shape and denatures it into its primary or secondary structure. That structure, when it touches your tongue on your taste buds, that tells your brain, hey, I'm tasting something differently, because that shape touches another shape on your tongue, your taste bud receptors, which goes to the neurons in your brain saying, you're not eating raw broccoli anymore, you're eating steamed broccoli. That's fascinating. That's also why if you ever have sushi to cooked fish, it tastes differently because the proteins are totally denatured when you cook the fish. So it depends on your taste preference. But if you eat mainly raw vegetables or raw fruit, it's going to taste different when you cook them. An apple tastes different than an apple pie, mainly because of all the sugar you're adding into it, but that the consistency, the texture, everything changes when you denature proteins. So denaturing proteins, so obviously the first way we could do this is by heat. So extreme amounts of heat can denature your protein. Now, if you go backwards, if you freeze proteins, you're not denaturing the protein. You're essentially just keeping it solid so it can't do its job. So you're preventing its catalytic activity. That's why we have refrigerators, that's why we have freezers, is because there are proteins in nature that love to decompose food and meat and everything we have, and we try to slow that process down by cooling it down. So that's one reason you want to do the opposite for that. So heat is one way. Another way, and this one's fun, is when you actually change the pH on something. So the pH is your proton power. We talked about this a little bit. The lower the pH, the more acidic something is. So have you ever, there's a beautiful biology lab on this that is easy to do and fun to do. If you ever cut an apple or an avocado, those are quick. If you ever cut an avocado or an apple and they kind of get brown over time, or potatoes, they kind of get brown over time. Have you ever done it and then added a little bit of lemon juice? They don't get brown as fast if you put some lemon juice on there. With the lemon juice, you have a lot of citric acid going on in there. So the acidic nature has basically denatured the proteins that are turning the fruit brown. And because as soon as you cut into fruit or anything like that, the fruit thinks it got injured. It actually did. You sliced it open, so it got injured. And all these proteins that are involved with injury repair start turning it brown to protect it. It's fascinating. So what happens when you put some lemon juice on a freshly cut apple, you're preventing those injury repair enzymes to do their job. So you don't let them go brown anymore. So changes in pH, and this could also be basic as well. So if you put, which I don't recommend, but ammonia and stuff like that would do something similar, but you can't eat that. So that's why we love to use lemon juice and stuff like that. So changes in pH. And I want you to think acid or acidic or basic. So heat, changes in pH. There's the other one that is very slight but very present in animal cells and plant cells and eukaryotic and prokaryotic cells is something called cofactors and coenzymes. So cofactors and coenzymes. And what you need to know is that these are essentially molecules that will bind to the enzyme to enable it to get the right shape to do its job. So if an enzyme is supposed to have, this shape, so it accepts something that looks like a circle into it, but it's naturally like this, what happens is a cofactor or a coenzyme is going to bind to the enzyme, which allows it to change its shape so it's actually functional now. And opposite can happen as well. You can have it in its functional state and a coenzyme or cofactor can bind to it and turn it off. So there's other ways to control the activity of an enzyme. All right, last thing we're going to talk about. You've been great. I know this is a longer lecture, but you're doing great. The last thing we're going to talk about is how we can block enzymatic activity. And big letter C, these are called enzyme inhibitors. These are huge in the pharmaceutical market. So enzyme inhibitors, we'll be quick. So enzyme inhibitors, they're naturally occurring. We turn enzymes on and off all day long, but the pharmaceutical market has learned to kind of hijack that same mechanism to target enzymes that are involved in bacteria or viruses or something like that. Everything that does something in cells uses enzymes to get their job done. If you can stop the enzyme, you stop the job. So normally, let's pretend to make an enzyme. I'll just make a very simple enzyme. All right, so this is enzyme. And this would be the natural substrate. Okay, so here's the substrate. All right, so this is normal. Okay, so this would be the normal situation. Let's say that this is a substrate that's involved in viral replication and this enzyme is involved in viral replication. I don't want the virus to work anymore in my cells, so if I block this enzyme, then the virus will essentially die out. So let's pretend to make some inhibitors. So this would be the normal case scenario. There's two types of inhibitors you can make. One of them would look like this. Like that. This is called a competitive inhibitor. Because it's actually competing for the active site. And now this natural substrate that's out here, it can't get in because it's being blocked. So it blocks the active site. And if this substrate can't get in, it can't produce the products it needs to do its job. So you kill its function. So that's a competitive inhibitor. The second kind, and you could probably guess, would be this guy. That right there is a non-competitive inhibitor. So what happened is this non-competitive inhibitor bound to the normal enzyme, but it bound to a spot that was away from the active site. And its binding caused a conformational change within the enzyme so drastic that it actually changed the shape of the active site. And now, when the substrate tries to come in, it can't because it has to be an exact fit. If it's not an exact fit, it's going to wobble and it's going to fall right out. It's not going to happen. So a non-competitive inhibitor changes the shape of the active site by binding